Biology 101. This is Amrita and let's start part 3 of the chapter Digestion and Absorption. Today's topic is digestion of food. In digestion of food, we have two types in our body, mechanical and chemical. And digestion starts in the buccal cavity and it has two processes, mastication of food with the help of teeth and tongue, then facilitation of swallowing. Mastication means chewing of food, facilitation means helping to swallow. Next, in saliva, we have mucus. So the two functions of mucus in saliva are lubrication and adhering masticated food particles into bolus. Lubrication means it makes the food slippery to make it easy to go into the stomach and esophagus and all. Then adhering masticated food particles into bolus means small food particles into a round like a small aggregate or something like that. Because with the help of saliva the small particles are made into bolus. Now bolus is swallowed and it goes through esophagus by successive waves of muscular contractions called the peristalsis. Now swallowing, another name of swallowing is deglutition, and in many question papers we can see deglutition as the word instead of swallowing. So remember that. And then through esophagus by successive waves of muscular contractions, that is called peristalsis. Now a peristalsis is a frequent word used in the previous years, so that is a word to remember and the definition as well. Next, coming to gastroesophageal sphincter, that controls the passage of food in stomach. What is gastroesophageal? From the word itself we can get the meaning. Gastro is stomach, esophageal is esophagus, and the esophagus is there, then the stomach is there. So in between that there is a sphincter, there is a guard. We have already learned many sphincters previously in this chapter itself. Now that guards the passage of food in the stomach. Next is saliva. What are the components of saliva? Electrolytes, enzymes, salivary amylase, lysosome. Now electrolytes are a sodium, potassium, chloride ion, then a bicarbonate ion. Then we have certain enzymes, then we have a salivary amylase, ptyalin, that's also called ptyalin, but this word is not there in the NCERT, but we can remember it if you want. Then a lysozyme, that is very important. Lysozyme, antibacterial agent that prevents infections. What component in saliva prevents infections? That is the lysozyme. Next, chemical process. We have learned now till this. There is a mechanical process. From them, then we have chemical process of digestion. In chemical process, this also starts in the buccal cavity itself. In buccal cavity, 30% of the starch is digested here. Whatever food we are taking, no, in that whatever starch is present, from that 30% of it is digested in the buccal cavity itself. Then in buccal cavity, hydrolytic action of the carbohydrate splitting enzyme, that is the salivary amylase. This is also called, for the salivary amylase, this is a bigger version of the name. So hydrolytic action of the carbohydrate splitting enzyme. They might say carbohydrate splitting enzyme, it's the same as the salivary amylase. Why do they call it as carbohydrate splitting enzyme? Because the 30% of starch is digested in the buccal cavity, right? In buccal cavity we have saliva. So that's why they have connected starch, carbohydrate. Now hydrolytic action means to break down, hydrolysis is to break down. So hydrolytic action of the carbohydrate splitting enzyme. Now this enzyme has a pH of 6.8 and starch to disaccharide, that is maltose. That in this pH only this reaction can happen. Starch in our mouth in the presence of salivary amylase in this pH, in this particular pH becomes disaccharide. That is maltose. Disaccharide example, that is maltose. Do remember that. Mucosa layer in stomach has gastric glands, and this gastric glands is in the form of three types of cells. First one is the mucous neck cell, second one is peptic or chief cells, third one is parietal or oxyntic cells. Now these three are very important to remember. Peptic or chief, parietal or oxyntic, don't confuse that. Then mucous neck cells secrete mucus. Peptic or chief cells secrete proenzyme pepsinogen. Parietal or oxyntic cells secrete HCl plus intrinsic factor, that is which helps, this intrinsic factor helps in the absorption of vitamin B12. These three functions are very important. This parietal part is an important PYQ as well. Now what is proenzyme, we'll come to that next part. Now coming to storage of food, the food in the stomach is present for 4 to 5 hours in our stomach, and then there it is mixed with acidic gastric juice, then it's churned. Churned means mixing, then it becomes a chyme. Now we already have learnt a term bolus. Bolus is when the food in our mouth mixes with the saliva and it turns into bolus. Now here the the bolus is mixed with the gastric juice. When it mixes with the gastric juice, it becomes chyme. So bolus, then to chyme, and then to chyle. Chyle is a word next to chyme, but that is not there in the NCERT, so no need to give much attention. Next comes the what is the proenzyme? We were talking about that, right? Now pepsinogen in the presence of HCl becomes pepsin. This pepsin is active, pepsinogen is inactive. This inactive form of the enzyme is called proenzyme. Before the enzyme, pro is before, so before the enzyme, that's why it's called proenzyme pepsinogen. Now pepsinogen is inactive in the presence of acidic hydrochloric acid, it becomes pepsin, that is active. Now what does this pepsin do? Pepsin in the presence, pepsin will make the protein to become proteoses plus peptones, that is peptides, together called peptides. So protein becomes proteoses plus peptones in the presence of pepsin. Pepsin is active form of pepsinogen. Now we also have a mucus, bicarbonates, and then renin. These three are present in gastric juice, and it also contains small amount of lipases. So read the textbook carefully. This page number 262 of NCERT of Digestion and Absorption chapter. It is you can read 1,000 times and there will be so many things you will be missing out on that page. So read that carefully many times. You will again and again find new points whenever you read it again. That is NCERT. So please read the textbook how many times you can. Then mucus, bicarbonates, some amount of lipases, and then renin. Now mucus and bicarbonates helps in lubrication, protection of mucosa from excoriation of HCl, because pH is 1.8, it is very acidic, right? pH scale from 1 to 14, in between 7 is neutral, 1 is very acidic, 14 is very basic, we all know that. Now pH 1.8 means it's near 1, that means HCl is very acidic. That means it, we all know, like HCl in the normal form in outside itself, it is very harmful to the human body parts. Now inside that's in the stomach itself we have HCl of 1.8, and it might be harmful to the inner surface of the stomach, right, the mucosa. We all have learned the 4 major layers of the alimentary canal. Now to protect that the mucosa from the HCl, we have mucus and bicarbonates present in the gastric juice. It also has renin. Renin, here the spelling is R-E-N-N-I-N. We also have another renin in the chapter excretion. There the spelling is R-E-N-I-N. So don't confuse in the question which renin they are asking. Now this renin is a proteolytic enzyme in infants to digest protein. What does this renin do? Like pepsin we have in adult body, right? Now this renin is the same proteolytic enzyme in infants to digest protein, to basically to digest the protein in the infant's body. That's what renin is. It's important, they might ask you. In bile we have bile pigment, bile salt, cholesterol, phospholipids, no enzymes. In pancreatic juice we have trypsinogen, chymotrypsinogen, procarboxypeptidases, amylases, lipases, nucleases. 6 of these. In intestinal juice we have disaccharide, dipeptidases, lipases, nucleotidases. These 4. So 6 plus 4, 10 things we have to remember. Bile, pancreatic juice, intestinal juice all go into the small intestine. Now, mucus plus bicarbonates from the pancreas protect intestinal mucosa, that is alkaline medium, pH of 7.8. This 7.8 for the enzyme to become active in the stomach. So, for that we have 7.8 pH, but this will be harmful for the inner layer, right? So, to protect that we have mucus and bicarbonate from the pancreas to protect the intestinal mucosa. Submucosal glands, Brunner's glands also help in this. So, these Brunner's glands also protect the intestinal mucosa. Now, all these in pancreatic juices are inactive enzymes. Already we have learned another inactive enzyme. Recall it, pepsinogen. Pepsinogen is the inactive form of pepsin, which gets activated by HCl. Now, intestinal juice, we have disaccharides, dipeptidases, lipases, nucleosidases. All these are enzymes. Whatever ends with ase, that is usually an enzyme. Even trypsinogen, all these. Now, next we have proteins, proteoses, peptones. These all change into dipeptides with the help of trypsin or chymotrypsin. Carboxypeptidase. These are pancreatic. We have learnt pancreatic juice. So, pancreatic trypsin or chymotrypsin. These help the proteins to change into dipeptides. Then these dipeptides become amino acids with the help of dipeptidases. So, see, actually in our NCERT they have given it in two different tables. That is, first only this they have given in a whole every whatever we have to change in our body, everything they have given one row. Then the second step another row, but I have written it together so that it will be easy to connect and learn. So, proteins, proteoses, peptones. Proteins become proteoses and peptones, right? So, this will become dipeptides. Dipeptides will become amino acids. So, dipeptides to amino acids, dipeptidases. ASS is there, so that is the enzyme which will digest dipeptides. This proteoses and peptones, from where it came? Before we learned protein to proteoses plus peptones in the presence of pepsin. So, that comes here. Then this with the help of pancreatic juice turns into dipeptides. Then from dipeptides to amino acids with the help of intestinal juice, that is dipeptidases. This is intestinal, this is pancreatic, so no need to confuse. Here 6 we have written, here 4 we have written, so we'll include all that. Now, why these proteins change into amino acids? I don't know if we have learnt it in smaller classes. All the proteins are made up of amino acid chains. Each amino acid, each amino acids like this will link into to form of protein. We have primary protein, secondary protein and all. So we'll learn that later. Actually, here in the intestinal juice, it is disaccharidases. It's not disaccharides. I just confused it and I wrote. I'm sorry. Now, next we come to polysaccharides. Same way, here proteins were converted, right? So, polysaccharides become disaccharides. Then these disaccharides become monosaccharides. So, polysaccharides, disaccharides, monosaccharides. Pancreatic amylase will be used on them to make it into disaccharides. Just they have given an example in the textbook: maltose, lactose, sucrose. All these are disaccharides. Now these become into monosaccharides with the help of these enzymes. Maltose, lactose maltase, lactase, and then sucrase. These three A's, it becomes maltose becomes maltase, lactose to lactase, and then sucrose to sucrase. So, all these are the enzymes. They become glucose plus glucose, glucose plus galactose, and glucose plus fructose. Maltose to glucose plus glucose to glucose, and then lactose to glucose plus galactose, and sucrose to glucose plus fructose. So, we need to remember this. This is in textbooks itself. In the old NCERT, it's 263 page number. but okay. So fats to diglycerides to monoglycerides, then these become fatty acids and glycerol. So fats to diglycerides in the presence of bile and or lipases. Now diglycerides to monoglycerides, then with the help of lipase again, it is fatty acids and glycerol. The process, no, it actually is given in two parts in the textbook, but we can learn it in one set itself so that it is easy to connect it. Now nucleic acids to nucleotides, with the help of nucleases, then nucleotides to nucleosides with the help of nucleotidases, nucleosides to sugar and base with the help of nucleosidases. So whatever we are converting, no, put an aces in between that, so that becomes the enzyme.